hi everyone. Can you guys please introduce yourself before we start the presentation one by one?
Hi, my name is Selvan Analaki Kasiraja. I am currently doing Bachelor in Computer Forensic. I am from Faculty of Engineering, Science and Engineering from FISIL.
That's good. Next.
Hi, my name is Mate Sharma, and I'm currently taking Bachelor in Games Design and Animation, and I'm from FISE.
Okay, next Maha.
Hi, my name is Mahalakshmi. I'm doing Bachelor in Graphic Designer and I'm from Faculty Physics.
Okay, that's good to know you guys. So I'm going to present one of the topic of that I learned during my this semester. So I hope you guys can understand well as what I learned. This is just for like share my experience with you about the medical knowledge. Okay, so I share my screen now. Can you guys can view my slide?
Yes.
All right. Okay. Thank you. So before we begin, I would like to explain you about the HPLC instrumentation. So basically, this HPLC known as a high performance liquid chromatography. In short form, we call as HPLC. So this HPLC known as the analytical chemistry used to separate, to identify, to quantify the component, the specific component that we want in any kind of mixture. So basically the HPLC that can be used is for like qualitative purpose. These are the qualitative purpose and also the quantitative. So basically qualitative is something like we call like we want to know the identification of the compound or the component on in the mixture. So basically, okay, if you go to if you go to any kind of mall to meet up your new friend or stranger that you want to like getting some parcel or some gift. So basically, if you want to identify them, you have to know their name, their address, any kind of their identity in the mall. Though, so in the mall, so that is the basically we call as the identify the person. So in the SPLC, we call like identify the component. Okay. For the quantitative is something like we call like okay during your cooking, you're adding some ingredient, right, for specific amount. So we call that as the specific quantity or quantity that you add on your food to ensure making a good present food. So that called quantitative. In medical, we use this to identify a component that comes into the mixture, either in food, cosmeceutical, or drugs. So basically, the drugs are formed by using various different kinds of ingredients actually. So basically, we can identify whether it's a present any specific ingredient that we want to know. For example, you want to identify if the drug is content of morphine or any kind of ingredient. So if you want to know whether it's present or not, you run this HPLC. In other words, or apart from that, if you want to know what specific strength or doses that using in the drug, you can compare in the HPLC as well by using the standard quantifications of that morphine. So these are basically that we are using the HPLC for identify and also to quantify. Hope we are clear with this basic principle of HPLC. Next, we move to how does it looks in real life. So this is the machine that looks like in our real life. So basically, there's a detector. Any kind of equipment or instrument, definitely there's a detector. As example, okay, if example you bringing car in the highway, right? So basically, it can detect whether you bring your car it's speedy or not by using the meter. Am I right? So basically, this instrument also works same as the car, whether the detector can show us the presence of the specific component that we're finding. And then there is the column chamber. Okay, the column chamber is something like we can call at most important item or the equipment or instrument for this HPLC, because without a column we cannot do any kind of separation of the component. Okay, and then there is a chromatogram. Okay, chromatogram is something that after we running this HPLC, we will obtain some kind of result. That's we call as a chromatogram, which is in shape of like there's a peak. There's a certain kind of peaks in the monitor. And then there is a sample manager where we do inject the samples and also the standard, and the solver manager. Solver manager, we always must be present there as a mobile face. Mobile face is something that can help you to flow the any kind of component that can be eluted. Okay, eluted means something that the component have been washed out from the HPLC. Okay, then okay, now we look into the working principle. Okay, in HPLC we have the one particular column that we using solvent. Solvent is something that can be moved, can be produced a movement or as a mobile. We call in another term or medical term we call as a mobile phase. Okay, mobile phase is something that can be moving, travel. Okay, can travel. And then there is a pump. Pump is to enhance the solvent to flow into the column. So basically the pump will be helping the solvent to go through into this column. Okay, and then there's the injector. Injector is of the sample or the standard that we want to inject into the HPLC. And then as I said before, column should be there for the separation purpose of the HPLC. If there's no column, there is no separation will be happen. And then obviously we have to be having the detector, and then after it being detected, it will be come out or it will be showed in the monitor via the chromatogram. Okay, and then after the component or the mixture had been detected, the wasted material will be gained into another container. So these are basically HPLC will be work, starting from the solvent. We inject the solvent and then we inject the sample, and then the solvent will be travel. The sample will be separated in the HPLC component due to the mobile phase and stationary phase, and then will be detected into the chromatograph, and then the non-used or the used waste will be get into another container. So okay, the phases that I'm meaning in this HPLC, there's two phases, which is stationary phase and also mobile phase. Stationary phase something that cannot be produced some kind of movement. It will be like static condition. And then the mobile phase is known as something that can be moved along, which can be traveled along the component or the mixture. Okay, as the image that I attached here, you can see here there is a slide and also the ball. So in this method, the slides are known as a stationary phase, which cannot be moved at all the time, but the balls on the surface are moving through downwards because it can produce the movement. So it's called as mobile phase. Now I can say that you can imagine that 50% of how the stationary phase and mobile phase work in HPLC. So basically the component that we want to identify by using the HPLC can be determined by, because there's varieties of the ingredient in the component that we want to inject, I mean the sample. So basically the ingredient that can be moved or can be same characteristic as the mobile phase, it can travel along with the mobile phase. If there's some ingredient that cannot move, cannot be attached to the mobile phase, it will be stayed at the stationary phase and it will become out later. So which means mobile phase will become fast and then followed back by the stationary phase component. So these are the how does it work and the needs of phase. If there's no phase, we cannot determine any specific or requirement that we need. Okay, so what are the one of the uses of HPLC is, for example, you want to determine some kind of ingredient. Okay, ingredient that present in any kind of food or drugs or cosmetic. Okay, so basically in this situation you're using a glucose, which are rotation time of 5 minutes as the standard, and also the sucrose, which is rotation time of 8 minutes. Okay, these are both are standards. So what you will be do, you will be run the standard, this both standard in the HPLC, and then determine the time of being eluted. As I said, eluted is something that washed out. Okay, so glucose being washed out at the 5th minute, and then the sucrose washed out at 8th minute. Okay, after you run the standard, standard is something that pure content which are comes with the one specific component. Okay, and then sample is means that you contain many kind of component inside the sample, so that they are showing many kind of peaks. So there's a lot of peaks that present a lot of a lot of components present in the sample. So basically this graph shows 3 peaks, so there's a 3 kind of component present in the sample. So what we're gonna do here, by using this standard and this sample, you can determine the presence of either the presence of glucose are there or sucrose. If you see this graph, at 2nd minute there is another peak, but which is not referred to 5th and also 8th. But if you see it at the 5th, you can see there's a peak which are presence of glucose. It can shows the presence of glucose. And but 8th minute there is no peaks, which that shows that there is no presence of sucrose. So in conclusion of this, we can say that by running the standard, we can determine whether the standard, the pure component are present in the sample as well or not by referring the retention time. If there is the same retention time as the standard, that means the standard are present in your sample as well. Are you clear, guys? Hope you guys clear. Okay. Okay, what are the parameters or the factors that can affect this HPLC? So basically, the first of all is the retention time. Retention time that I mentioned just now is something that can be traveled, something that can be traveled in the HPLC and being eluted. Okay, so as fast short the retention time, it means that it being eluted very faster, which will come out faster or first from the HPLC. That's very important for this HPLC. And also, retention is something that we can use for the identification. Just now I explained for you by showing the time, right? The 5th minute, you saw there's a presence of glucose. Am I right? So by running that, you also can determine the retention time, compare the retention time, and also you know the what are the specific identification of this component are present. Okay, the second thing is selectivity, which selectivity is something that can be differentiated between the 2 components. For example, this is the selectivity, right? So as far as the peak, it shows the best differentiate of the or the good resolution of HPLC. If these both peak are overlapping with each other, which means like too close, it means the components are not separated very well. It should be very far. For example, this and this are so far. Am I right? I mean, not even touch very close. So it's very good. Even for this also very far, very good. This also far, so very good. Okay, that's the one of the parameter. And then the third one is efficiency. Efficiency is something that the size of the peak in between this one. Okay. So the efficiency are very good when the peak are narrow, not too broad. If it become too broad or fat, it is not a good condition. You have to use some other kind of column in HPLC to enhance a good efficiency. If too broad, it shows like the peaks are like very low, and also the space are very big. So it's not good for the HPLC reading, and also the spread of data will be not that much good. Okay, so these are the main 3 factors or parameters we have to know in HPLC. Okay. Next is Next, I will talk on to the examples. If you see this example, the first one, this shows is better selectivity. Am I right? Because it's like too far, better selectivity, and also a good efficiency. Am I right? Because it's not too fat. Am I right? It are too narrow. So it's good efficiency and also good selectivity. As for the second one, these are, I will say, like poor selectivity, but good efficiency, because these are not too broad, very narrow, good one, good efficiency. But if you see the selectivity, these are like slightly too close. Am I right? So if it's too close, the selectivity are very poor. It's not like very good for the identification. For the third sample, the peaks are too broad. So this shows like good selectivity because it's like far a bit, but the efficiency of the each peak are not that good because it's a fact. It's like too broad. It should be like narrow like this. So it can like say like less efficiency. And for the fourth image, this shows overlapping. So if overlapping, it's not like too not far, it's like very close. It shows very poor selectivity, and also these things are very broad, shows the poor efficiency, which are very not good data that being gained. These are good data and good result. Okay. Until now, I hope you guys can get it. What I mean, this and these are the overall image that you can see in the HPLC and what you think happens in the HPLC. Hope you guys are clear. So if you guys have any questions, can shoot to me. I will try to answer you. If you guys can't understand anywhere or any part of the explanation, I'm willing to explain you right now or maybe later, but you can still ask me the questions lah. Doesn't matter. I hope you guys are clear about this thing lah. If you guys not understand, you guys are required, or you guys can ask me to repeat the thing again. I have no problem. So guys, any questions?
Hi, Tarvin.
Yes, Shelvan.
Your presentation was very nice,
Thank you.
and your slides were very good. Even I'm not a science student, I'm able to understand the concept of the qualitative, quantitative, and how the machine works, and the HPLCs detector, how the detector work with, and also the factors all. And your explanation was very nice, and the slides look very simple, and we all can understand about what you try to say with us. And I have a question about the slide number 4, which is the faces.
Yes.
Why there is different faces? Can you explain more about it?
Okay, can. Doesn't matter. Okay, basically in HPLC, there's two phases that I said before, which is mobile phase and also the stationary phase. Okay, if there is, okay, as the column is a stationary phase, okay? If that thing is stationary phase and the component that you want to identify, there's a different kind of component. So basically, some component can be stayed at the stationary phase as they're having the same criteria as the stationary phase. But some of the component really cannot be attached at the stationary phase because some can be travel along. So basically, we need mobile phase as well to ensure the component that attached to mobile phase can be eluted first, and also the stationary that the component that attached to stationary phase eluted later. So we can determine by that, we can determine which are very good or which are the very present in like shorter time, and also which are very late come out, and also like we can determine that the component are like non-polar or polar. We can determine the specific character of them. So basically we need both of them, both of the phases to enhance the good performance. Yes, because different kind of component having different kind of polarity, which means can be attached to mobile phase, which some can be attached to stationary phase. So if you have one kind of phases, there's nothing like some, I mean like all of the component will be at one place, so cannot be separated. That thing can, the component that we want to be separated cannot be separated because having some kind of different shape. Am I right? So that's the thing. If for example, I'm showing you some example. During you cutting your, during cooking, you are cutting onion, right? So you're using the cutting board. Am I right? The cutting board is a stationary phase. The knife that you're using during the cutting is a mobile phase. Why I'm saying that, we thought that cutting board and also the mobile face, the knife, you cannot properly cut the onion. Am I right? So basically, you need both of them in order to get the good separation. Yeah. Am I answer your questions?
Thank you for the explanation, Tarvin. I
Okay.
understand now.
Thank you. Thank you
Thank
so
you so
much.
much.
Questions. Very good questions. Any other?
Okay, I have a question.
Okay. Yes. Yes. Ma'am.
Okay, question about the samples.
Samples. Yes.
Okay, if the samples doesn't match the standard as you said, what will be the solution?
Okay. What you mean is like the sample, the unknown sample that we want to determine the presence of component which are not same as the standard. So these are most probably referring to the identification. So here the solution, we can say that you have to make a proper standard sample or standard solution, or testing the good standard that you are using before you are running your sample. So what I'm meaning here is the standard that you choosing should be studied first very well, whether it can be present or not. You cannot simply cannot simply select a standard and just run in this PLC. You have to know or identify or research about it, whether the standard can be present in your sample or not by referring to the samples. So you have to study both samples and also the standard, which can be compatible. After you know that, then only you can select your standard, and then you run in the HPLC, and also can be run your sample, and then the component can be same or match as the standard solution. So the basically the solution can be given is studying or research properly before you selecting your standard. You must know what kind of standard you have to be using to for the specific component that you're finding. So that will be best solution. Yes. Yes, Matish.
Okay, thank you for the answer.
Okay, thank you. Thank you, Matish, for the questions. Any other questions? Welcome.
Hi, I have a question.
Yes, yes, Ma, yes.
What to do when there is no pics?
What we will do where there is no peaks, which means the chromatogram doesn't show any kind of peaks. Okay, that was very good questions. Okay, if there's no peak, it's mean like the sample that we injected cannot be determined by the HPLC. There is some kind of major problem, or the method that we are doing, or the analyzer that inject the sample doesn't do the procedure very well. Or maybe we can say that the mobile phase that I mentioned before, the solution or the solvent that using to determine the separation, are very wrong mobile phase that we're using. Okay, here we can say for determination of the component also should be same and compatible with the mobile phase that we're using. If you're choosing that something that cannot be interact or cannot be run the specific component, so that will be no peak shown. So which means the mobile phase cannot bring along the component that want to be separated. It must be same characteristic or criteria that can be having same as the mobile phase. And also we can say there is some kind of blockage in pump. If the pump are broken, the solvent cannot be flow into the column, as the mobile phase cannot be travel or cannot be separate the specific component. So these are kind of problem of the individual itself, equipment itself, the sample itself, and also the person in charge itself. Yes. So if you wanted to fix this, you have to run it maybe around 3 times, or with a different kind of person or different kind of analyzer, to ensure the process are running in good, or to avoid any kind of minimal error. Yes. Yes. I hope I answered you. Any
Yes,
kind
thank
of,
you.
all right. Thank you, Ma, for that question. Very good questions from you guys. Is there any questions?
No, Tarvin.
Okay, if no means I'm really I'm really happy to teach you guys about this HPLC. And if in future, if you guys want to know more about HPLC, actually there's more to learn about HPLC. I can tell you, but maybe in future, if you want to interested, this one is I just explained to you briefly, as you can imagine what is happening in the industries and how can we determine a specific component that we want to know in the some kind of drugs, products, and things. Okay, so that's all I want to share with you. If there's no questions, I hope you guys can help me by scanning this QR code that I had been shown here in the slides and can be evaluate me honestly. Doesn't matter. You just evaluate me, my presentation, and also my quality of explanations. Thank you guys. Can you guys scan?
Can.
All right. So if you can, thank you guys. Thank you for your times. I'm really appreciative. All the best for our future. Thank you.
Thank you, Tarvin.
All right, thank you.
Thank you too.
Thank you, Matesh. Thank you, Salman. Thank you, Ma.